Hello students, a warm welcome to all of you in our 3 days online certification course on topic molecular docking. In today's session, we discuss about the overall idea of molecular docking, what is molecular docking, how we prepare the protein, how we prepare the ligands for the docking, and we also discuss how we predict the tertiary structure of the protein by the help of AlphaFold. Okay, so we start today's session and understand what is molecular docking. Okay. So, what is molecular docking? Molecular docking is a computational method. What is molecular docking? Molecular docking is a computational method that are used to predict how the ligand is bind to the receptor. Okay, your ligand is your small compound. Okay, and your receptor is your biological target. Okay, this is your ligand and this is your target. Now, how this ligand is bind with this target and form this complex, this interaction we study by the help of computational tool, that is the molecular docking. How this particular ligand is bind to this receptor. Okay, basically we find this result in a form of affinity. Okay, less the value, we find the result in a form of energy. Less the energy, less the affinity, more the binding. Affinity is inversely proportional to the binding. More the binding, less the affinity. Okay. Now, molecular docking is a computational method that are used to predict how the small molecules bind to the target protein or receptor. Okay. By the help of protein, we easily determine the binding orientations. It means how the ligand fit inside the protein with active site. Let's suppose this is the active site of this binding biological molecule. So by the help of molecular docking, we easily understand what in which pose this particular ligand is bind to this protein or this biological receptor. And we also find the binding affinity. Okay, how the strongly ligand bind with your protein, and that this result is also known as a binding energy or docking score. Less the docking score, more the affinity. Okay, it is inversely proportional to each other. Next, protein and ligand interactions. For protein and ligand interaction, we easily find out how the protein in which next is your protein and ligand interactions. Means whenever the ligand is bind to your protein, then how many interactions will work, like hydrogen bonds, hydrophobic contracts, ionic bonds, and pi-pi stacking and pi-pi stacking. These are the interactions. Okay, so for as molecular docking is a computational tool, so in bioinformatics, number of docking softwares are available like Autodog Pyrex, Clespro, Sysdog, HeadDog, Patchdog, TVDog, and your MU. Okay, These are the software as well as web tool to perform the molecular docking. Okay, Autodogbina, Pyrex, MOV, it is a software that are used to predict the protein docking. And this Clestro, SysDoc, HeadDoc, PestDoc, CBDoc, it is a web tool to perform the molecular docking. Okay. Whenever we perform the docking, then whenever we perform the docking, then mhm. whenever we perform the docking, then we find the result in a form of scoring value. Less the scoring value, more the affinity. Okay. So now we understand why this molecular docking is so much important. Okay. Here, this is the in this figure, this is clearly mentioned. This is the active site of your protein. This is the active site, and this is your ligand, and this is the 2D structure, 2D graph of interactions. Means this is the amino acid, this is the ligand is bind to this amino acid of your target protein, and this is the 3D view or 3D view of ligand and protein interaction, and this is the side view. Okay, okay. This is the different representation of your docking result. Okay, this is your active site. This is the complete. This is your active site. This is your ligand. Now ligand is put on this protein, and in this protein, and this ligand is bind to these amino acids of this particular protein, and this is the 3D interaction of protein as well as ligand interaction, and this is the 2D structure of ligand and protein interaction, and this is the side view. Okay, now we understand the importance of molecular docking. Molecular docking is widely used in drug discovery. How? First, in drug discovery is based on two approaches. First is your ligand based, second is your structure based. Okay, let's suppose you know, you already know about the drug. Okay? Now this drug means your ligand is known. Here the ligand, your drug is act as a ligand. Okay. Now by the help of different software, we submit that drug and find out what are the biological target of this particular drug. Okay. And selecting this particular biological target, then we perform the molecular docking. Okay. After the docking, we easily find out with this particular drug or this particular ligand, how the after the docking, we easily find out what is the affinity between this drug as well as our biological target. How strongly this drug is bind to our biological target. Then it is also helpful to discuss in detail about all these points. Okay. Now, what is the core principle? Core principle is better the fitting of the ligand means more stable complex. Lower the binding energy, strong, longer the binding energy, strong the binding. Okay, means lower the docking score, higher the binding. Of the higher binding means your stable, your structure is more stable. Now we discuss about the key concept of docking. First is your receptor. What is your receptor? Receptor is your biological target. Biological target where the ligand will bind. This is a this provides the binding site for ligand. It provides the binding site for the ligand. Okay. This ligand is bind to the receptor by different interactions like hydrogen bonding, hydrophobic interactions, etc. Okay. Most of the time your biological target is protein. But sometimes this biological target may be your DNA, maybe your RNA, maybe your enzyme, maybe your any receptor. Receptor like GPCR receptor, tyrokinase receptor, ion channel receptor, etc. Okay. Now, whenever we select the receptor for the molecular docking, there are some criteria which we consider or which must be considered. What the criteria? Criteria is your protein is properly folded. Your protein must be properly folded and we clean this protein. Next step is your cleaning. Yes, your protein should be clean. Clean means no water molecule, no extra chain. If we perform the specific docking, if we perform the blind docking, there is no issue. And no heteroatoms, etc. Only your macromolecules is present, means pure chain of your protein is present. Okay. So before the molecule docking, we clean the structure by the help of different software like PyMol, Discovery Studio, etc. Okay. Then we add the Coulomb charge. Okay. And this charge is very important for performing the molecular docking or autodock. Okay. And this receptor provides the binding site for the receptor, for the ligand. Okay. It provides the binding site. Now your ligand is bind to this binding site and perform their task. This is your receptor. Okay. Now we discuss about what is the Coulomb charge. What is the Coulomb charge? Coulomb charge are partially charged computed by fitting the quantum mechanism calculator. Calculated electrostatic potential around the molecule. By the help of this charge, software understood how the electrons are distributed in the molecules, how the molecules will interact with electrostatic and receptor or other molecules, and how strong hydrogen bonds or ionic interactions. Okay. Now, these Coulomb charges are usually assigned to the proteins. Okay. Whenever we prepare the protein for the docking purpose, then we assign this charge, that is the Coulomb charge. Why we add this charge? Because protein structure do not contain the charge information. Okay. Docking algorithm need to charge to calculate the electrostatic interactions, binding affinity and orientation of ligand. That's why we add this Coulomb charge before the docking. Okay, this is the importance of this Coulomb charge. Okay. Now we discuss the next point, that is the ligand. What is the ligand? Ligand is a small molecules. What is the ligand? Ligand is a small molecule that will bind to the receptor and perform their task. And your ligand may be any drug, maybe any inhibitor, maybe any biological compound, maybe the peptide. Okay. And this ligand is bind to the active site of protein and perform their task. Okay, basically whenever the ligand is bind to the receptor like GPCR receptor, then it will start the signaling process. Okay. Okay. And before the docking, first we prepare the ligand. For preparing the ligand, first we minimize the energy, then protonated form, then assign the next we assign the Castigar charges. Okay. Then we convert the ligand in PDBQT format. Now our ligand is ready for the docking purpose, for the docking. Okay. Now, next is your binding pocket. This is your binding pocket. It is also known as a cavity. This is a cavity where the ligand will bind. Okay, this is a cavity, and these are the amino acids which are showing the binding affinity with our ligand. Okay, now what is the binding pocket? Binding pocket is a 3D cavity or group of protein where the ligand is bound. It contains the catalytic residues, hydrophobic pockets as well as metallic ions. Okay, this pocket determines the selectivity, binding strength as well as molecular interactions. Okay. Simply binding pocket is a site where the ligands will bind. Okay. Now scoring functions. Means what is the scoring functions? Because we find the result in a form of scoring value. And this scoring value is a mathematical formula that are used to estimate the binding affinity. We find the output result in a binding energy in kilocalorie per mole. Negative the value, strongly the predictions. Okay. Why the scoring? What scoring functions measure? It will measure the hydrogen bond, hydrophobic interactions, electrostatic interactions, van der Waals interaction, dissolution energy, and total energy. Okay. By the help of a scoring function, we easily find out all these energy. Okay. And these are the different scoring value like autodog scoring, Vena scoring, glide score, or gold score, CBD gold score, etc. These are the different scoring value which we estimate after performing the molecular docking. Okay. Now we discuss about the docking workflow. Okay. What is the docking workflow? First step, we predict the protein or retrieve the protein. For retrieving, we use a database like your PDB, like UniProt, like AlphaFold. Okay, okay. Then next step, after downloading this protein in PDB format, then we perform the cleaning part. In cleaning process, we removed out the bottom molecule, heteroatoms, etc. Okay, and also removed out the unessential cofactors. Or if in our protein ligand is present, then we also remove the ligand. Okay, for this, for removing all these points, we used a software that is the Pymol as well as Discovery Studio. After cleaning the protein, then next step is your protein preparation. For protein preparation, then if we perform the molecular docking by autodog DNA, if we perform the molecular docking by the help of autodog DNA, then we load this protein on autodog DNA, then add the polar hydrogen molecule, also assign the Coulomb charges. Okay, then convert this into the PDBQT format. Okay, now whenever the protein is converted into the protein PDBQT format, now our protein is prepared. Then next step, ligand preparation. Okay. For ligand preparation, if our ligand is already present in database like PubChem, then we download the ligand in SGF 2D structure. Okay. Then open this, and if our protein or our ligand is unique, then we draw the structure on ChemSketch on OpenWebel. Then we scratch our ligand on ChemSketch, or we also on PubChem, etc. Then open into the OpenWebel. Then OpenWebel is a software that are used to change the format. Okay. Then we open it into the Discovery Studio, and in Discovery Studio we convert this 2D structure into the PDB format. Okay. After downloading this ligand, then we prepare this ligand. For preparing the ligand, first we add the hydrogen molecule, then minimize the energy. If we perform the molecular docking by the help of photo dock Venus. Okay. Then assign the gesture charges or define the rotamer bolts, and finally convert the ligand into the PDB QT format. Okay. Now our ligand is prepared. Our ligand is prepared as well as our protein is also prepared. Then we move to the docking portion. For docking portion, we also use Autodogbina, Autodogbina 4, Docthar, Glad, Slide, SDoc, SysDoc, CBDoc, etc. Okay. Then we finally then docking procedure means multiple pores by find the binding energy or orientation of ligand. Okay. Then finally we find the result in a form of scoring value. Less the scoring value, more the activity. And then finally we analyze the protein and ligand interactions in different software like Discovery Studio, PyMol and LinkPlot. Okay. And by the help of on this software, by the help of this on this software, we easily check the hydrogen bonding, hydrophobic interaction, pi-pi interactions, and salt bridges. Okay, and now our protein and ligand complex is ready for the simulation part. Okay, first step is your protein and ligand preparation, protein and ligand downloading. First you download the ligand as well as protein, then we prepare the ligand and the protein for the docking purpose. Then we perform the docking. After performing the docking, we find the different poses and different orientation. Then we on the basis of scoring value, we predict our result. Then we move to the further analysis, means we check the 2D graph, 3D graph, and their interactions, etc. Okay, and this is the complete workflow of molecular docking. So this is all about, this is the overall idea of molecular docking. Now we switch to the practical portion. In practical portion, first we understand from the molecular docking by the help of Pyrex. Then we visualize the result of Pyrex in Discovery Studio.
Thank you so much. In next lecture, we discuss about molecular docking in detail. Thank you.